which require the surface. Most of the cells which require the surface, like um, an artificial substrate like adherent or monolayer culture, whereas other can be grown in a free floating culture medium called suspension culture. Depending upon their lifespan of the cells, most of the cells is genetically determined, but some cells, cell culturing cells have been transformed into immortal cells. Those immortal cells which will reproduce indefinitely if the optimal conditions are provided. We also discussed about the primary cell cultures. Primary cell cultures, here we will discuss about primary and secondary cell cultures. A primary cell culture which is obtained from the animal or plant tissue, and the secondary cells which is originated from a, a primary cell culture closely resembles the parental tissue, and secondary cells are not closely resembles to the parental tissue. The biological response of the cell may be closure, that an in vivo environment. The in case of secondary cell culture, the biological response of the cell differs from that an in vivo environment. The first cultured cells are derived from original cells, tissue from an in vivo environment are called primary cell culture, whereas the cells which is derived from an already existing culture is called as secondary cell culture. And primary cells that cannot be transformed, secondary cells can be transformed. Primary cells having the less chance of mutations, whereas secondary cells increase the chance of a mutation or genetic alteration of primary cells. Uh, primary cell Primary cell culture acquired through steps of rinsing, dissection, and mechanical or enzymatic disintegration, whereas in secondary culture, if the primary culture is an adherent culture, the first step to the detachment of the cells from the attachment to the surface vessel by mechanical or enzymatic means of separation. Then the cells have to be detached from each other to form a single cell suspension. Here in the primary cell culture, finite lifespan. In secondary cell culture, prolong the lifespan of the cells, and periodic subculturing may produce immortal cells through the transformation or genetic alteration of the primary cells. And the risk of contamination is very high in case of primary cell culture, whereas in the secondary cell and also difficult to maintain, whereas in case of secondary cell culture, the risk of contamination is very low and comparatively easy to maintain. And uh, in the past lessons, we already discussed about the what is cell line and what is cell stem. But still, here, before going to the next chapter, once a primary culture is subcultured or passaged, it represents a cell line. And whereas a cell line is permanently established cell culture which will proliferate from if a suitable fresh medium is provided continuously, whereas cell lines have been adapted to the culture, but unlike cell lines, have an infinite division of the potential. Cell strain is obtained either from primary culture or a cell line. This is done by selection or cloning of those particular cells having specific properties or characteristics which must be defined. And isolation of cells, as just we discussed about the primary cells, how we can isolate the cells means firstly we have to digest the tissue with the enzymes called collagenase, trypsinase, pronase, and before agitating the tissue to release the cells into the suspension. Alternatively, pieces of tissue can be placed in the growth media, and the cells that grow out are available for culture. This method is known as explant culture. Cells that are cultured directly from a subject are known as primary culture. And then how to maintain the cells in the culture medium. We also discussed about the culture medium. For the majority of the isolated primary cells, they undergo the process of senescence and stop dividing after a certain number of populations doubling, which generally retaining their viability called Hayflick limit. Cells are grown and maintained at an appropriate temperature, and the gas mixture is 37 degrees centigrade and 5% CO2 for mammalian cells in a cell incubator. And culture conditions vary widely for each cell type, and variation of conditions for a particular cell type can result in different phenotypes. Aside from the temperature and gas mixtures, the most commonly varied factor in the cell culture systems is the cell growth membrane. Cell growth medium. What are the recipes for the growth media can vary. pH, glucose, and concentration growth factors, and the presence of other nutrients like the growth factor used to supplement media are often derived from the fetal bovine serum, bovine calf serum, equine serum, and porcine serum. One complication of these blood derived ingredients is the potential for contamination of the culture with viruses, prions, particularly in medical biotechnological applications. And what is plating density? The number of cells per volume of the culture medium. It plays the critical role for some cell types. For example, a lower planting density makes granulosa cells exhibit estrogen production, while a higher plating density makes them appear as progesterone producing theca lutein cells. And what are the components of the cell culture media? First one is carbon source, that is glucose and glutamine. This is the source of energy, and amino acid, which is the building blocks of protein, and vitamins promote cell survival and growth. A balanced salt solution, an isotonic mixture of ions to maintain optimum osmotic pressure within the cells and provide essential metal ions to act as cofactors for enzymatic reactions, cell adhesions. And phenol red, pH indicator, the color of phenol red changes from orange red at pH 7 to 7.42 and yellow at acidic a lower pH and purple at a basic higher pH. Bicarbonate buffer, it is used to maintain a balanced pH in the medium. And the main thing is temperatures. For the typical growth conditions, temperature 37 degrees, CO2 5%, and relative humidity 95%. And cell line cross contamination to address this problem of cell line cross contamination, research are encouraged to authenticate their cell lines at an early passage to establish the identity of the cell line. Authentication should be repeated before freezing cell line stocks every 2 months during the active culturing, and before any publication of research data generated using the cell lines. Many methods are used to identify the cell lines, including isoenzymes analysis, human lymphotic uh, antigen typing, chromosomal analysis, karyotyping, morphology, and STR analysis. What are the technical problems associated with cell culture? as natural as cells generally continue to uh, divide in culture, they generally grow to fill the available area or volume. This can generate several issues like nutrient depletion in the growth media, changes in pH of the growth media, accumulation of apoptotic, necrotic or dead cells, and cell to cell contact can stimulate cell cycle arrest, causing cells to stop dividing, known as contact inhibition. Cell to cell contact can stimulate cellular differentiation. Genetic and epigenetic alterations within natural selection of the altered cells, potentially leading to overgrowth of the abnormal culture adapted cells with a decreased differentiation and increased proliferative capacities. And hmm, manipulation of cultured cells. Among the common manipulations carried out on culture cells are media changes and passaging the cells and transfecting the cells. These are generally performed using the tissue culture methods that rely on aseptic technique. Aseptic technique aims to avoid contamination with bacteria, yeast, or other cells and other cell lines. Manipulation are typically carried out in a biosafety cabinet or laminar airflow cabinet to exclude um, to avoid the contamination and exclude the contamination with the microorganisms, and the antibiotics like penicillin, streptomycin, and antifungal agents like amphotericin B and antibiotic antimitotic solution can be added to the growth media. As the cells undergo metabolic processes, acid is produced and the pH decreases. Often a pH indicator is added to the media to measure nutrient depletion. What are the media changes we can observe is in the case of adherent cultures, the media can be removed directly by aspiration and then is replaced. Media changes in the non-adherent cultures involve centrifugation, the culture and resuspending cells in the fresh media. And transfection and transduction. Another common method for manipulating cells involve the introduction of the foreign DNA by the transfection. This is often performed to cause cells to express a gene of interest. More recently, the transfection of RNAi constructions have been released as a convenient mechanism for the suppressing the expression of a particular gene or protein, and DNA can be inserted into cells using viruses in methods referred to transduction, infection or transformation viruses as parasitic agents as well as suited to introduction DNA into cells, as this is a part of their normal course of reproduction. And what are the applications of cell culture? This is a special synopsis on tissue engineering and vaccine production. Mass cultures of the animal cells is fundamental to the manufacture of viral viruses, viral vaccines, and other products of biotechnology. Culture of human stem cells is used to expand the number of cells and differentiate the cells into various somatic cell types for transplantation. A stem cell culture is used to harvest the molecules and the exosomes that the stem cells release from the purpose of therapeutic development. Biological products produced by the recombinant DNA technology in animal cell cultures, including enzymes, synthetic hormones, and immunobiological like monoclonal antibodies and interleukins and lymphokinins and other anti-cancer agents. Although many simpler proteins can be produced using the RDNA technology in the bacterial cultures, more complex proteins that are glycosylated, means carbohydrate modified one, currently must be made in animal cells. And another important example of such a complex protein is the hormone erythropoietin. The cost of growing mammalian cell culture is high, so researchers are underway to produce such complex proteins in insect cells or in higher plants. Use of single embryonic cell and somatic embryos as a source for their direct genetic transfer, that is genetic direct gene transfer via particle bombardment technique, transit gene expressions and confocal microscopy observation is one of its applications. It also offers to confirm single cell origin of somatic embryos and the asymmetry of the first cell division, which starts the process. A cell culture is also a key technique for cellular agriculture, which aims to provide both new products and new ways of producing existing agriculture products like milk and cultured meat and fragrances and rhino horn from cells and microorganisms. It is therefore considered one means of achieving animal-free agriculture. It is also considered like an animal-free agriculture. It is also a central tool for teaching the cell biology techniques. When coming to the tissue culture and engineering, cell culture is a fundamental component of the tissue culture and tissue engineering, and it is establishes the basics of growing, maintaining cells in vitro. The major application of human cell culture is in stem cell industry, where mesenchymal stem cells can be cultured and cryopreserved for further use. Tissue engineering potentially offers dramatic improvements in low cost medical care for hundreds of and thousands of patients annually. Vaccines. As we know, polio vaccine, measles, mumps, rubella, and chickenpox are currently made in cell cultures. Due to the H5N1 pandemic threat, research into using a cell culture for influenza vaccines is being funded by United States government. Novel ideas in this field include recombinant DNA based vaccines, such as one made using human adenovirus, a common cold virus, as a vector, and novel adjuvant. The next topic is, this is about the complete animal cell culture and media, and what are the media components, and cell culture media, plate density, and how to maintain the cells in the culture, and isolation of the cells. This is finding that the concept in mammalian cell cultures are finished. And our next topic is stem cell. Stem cell and their applications. So here stem cells. First we have to know about the stem cells. The first one is what are stem cells? What are stem cells here means? Stem cells are raw cells found within the human body from which all other cells with specialized functions are generated in proper experimental conditions. These stem cells can be divided to create daughter cells. These daughter cells can either be new stem cells themselves by way of self renewing, or end up being functional cells. So, for example, blood cells or brain cells. And then, where do these stem cells come from? Our research have discovered several sources of stem cells. Before going to that, again, what is stem cells? Stem cells are undifferentiated. A simple definition for the stem cells are undifferentiated biological cells that can differentiate into specialized cells or can divide to produce more stem cells. They are found in multicellular organisms. In mammals, there are two types of broader types of stem cells. One is embryonic stem cells and embryonic stem cells, which are isolated from the inner cell mass of blastocells, and adult stem cells, which are found in various tissues. In a developing embryo, stem cells can differentiated into all the specialized cells called ectoderm, endoderm, and mesoderm. Here, embryonic stem cells. These embryonic stem cells come from embryos that are 3 to 5 days old. They are by far the most versatile and can thus be used for both regenerative and cell replacement process. And when coming to the adult stem cells, these stem cells, adult stem cells, these stem cells, which are found in various tissues, um, stem cells are found in most adult tissues, such as bone marrow or fat, in small numbers compared with embryonic stem cells. These have a more limited ability to produce various cells found in body. These can be versatile but not to extent found in embryonic cells. And then and before going to the futures of the stem cell, adult cells altered to have properties of embryonic stem cells. Most of the scientists have successfully transformed regular adult cells into stem cells using genetic programming, genetic reprogramming, through altering the genes into adult cells. Researchers can be reprogrammed the cells to act similar to that of embryonic stem cells. And perinatal stem cells, researchers have discovered stem cells in amniotic fluid as well as within umbilical cord blood. These have the ability to change into specialized cells where required. And what are the features of stem cells? Here the stem cells are unique, very unique cells. Stem cells have the amazing ability to develop into several distinct cell types in the body. Stem cells can be used as a repair system for the body. The stem cells can divide without limiting in a living organism in order to replacing the various types of cells. When a stem cell divides, each new cell has the potential to either remain a cell, a stem cell, or becoming another type of the cell with a more specialized function, that is a muscle cell or a red blood cell or a brain cell, etc. As just we discussed, the classification of stem cells based on the origin. Stem cells from the embryo, called as embryonic stem cells, which is also short termly called as ESCS. And other one, The other one, stem cells from fetus, stem cells from umbilical cord, and stem cells from adult. Depending upon the cell type, the stem cells are derived like this. First of all, embryonic stem cells. Embryonic stem cells are pluripotent stem cells are derived from the inner cell mass of a blastocyst, an early stage pre-implantation embryo. Human embryos each the plastocell stage 4 to 5 days post fertilization, at which time they consisting of 50 to 150 cells, isolating the embryoblast. 1 second. Ah, Isolating the embryoblast stage of embryoblast or inner cell mass, resulting in the destruction of the blastocyte, a process where raises ethical issues, including whether or not embryos at the pre-implantation stage should have the same moral consideration as embryo in the post implantation stage of the development. Then what are the properties of embryonic stem cell? Is the embryonic stem cells derived from the blastocyst stage of early mammalian embryos are distinguished by their ability to differentiate into any embryonic cell type, and by their ability to self-renew. It is these traits, these traits that make them valuable in the scientific and medical fields. These embryonic stem cells have a normal karyotype, maintain high telomerase activity, and exhibit remarkable long-term proliferative potential. So first one is pluripotent, and the other one is self-renewing and a repair of structure and growth. Uh, Growth of the embryonic cells divide very frequently due to the shortening of G1 phase in their cell cycle. A rapid cell division allows the cells to quickly grow in number but not in size, which is important for early embryo development. And in this embryonic stem cells, um, cyclin A and cyclin E proteins involved in the G1 and S transition stage of the cell cycle are always expressed at high levels, and CDK2 that promote cell cycle progression are overactive, in part due to down regulation of their inhibitors. Retinoblastoma protein that inhibit the transcription factor E2F until the cells is ready to enter S phase are hyper phosphorylated and inactivated in this embryo stem cells until cell is ready to enter into the S phase and hyper phosphorylated and which leads to continual expression of the proliferation genes. These changes resulted in accelerated cell cycles of cell division through the shortening of G1 phase has been linked to maintenance of pluripotency and growth in this embryonic stem cells grow in serum free conditions do express hypophosphorylated active RB proteins and have an elongated G1 phase. The pluripotency factors play a role in transcriptionally regulating the embryonic stem cell cycle. Then what are the uses of doing all these? Embryonic stem cells means already we discussed about the self renewal and renewal and what we discussed um, um, what is that embryonic stem cell therapies have been proposed for regenerative medicine and tissue replacement after the injury or disease. Pluripotent stem cells have shown promising results in treating a number of varying conditions, including but not limited to the spinal cord injuries, age related muscular degenerations, and diabetes and neurodegenerative disorders such as Parkinson and AIDS, etc. In addition to this, to their potential regenerative medicine, uh, this embryonic stem cells provide a possible alternative source of tissue organs. These embryonic stem cells also be used for research on early human embryo development, certain genetic diseases, and in vitro toxicology testing. Human embryonic stem cells have the potential to differentiate in various cell types and thus may be useful as a source of cells for transplantation or tissue engineering. And what are the clinical potency of this um, embryonic stem cells are? Embryonic stem cells have been differentiated in natural killer cells and bone tissue. Researchers have differentiated these embryonic stem cells into dopamine producing cells, with the hope that these neurons can be used in the treatment of Parkinson disease. There are studies which involves in embryonic stem cells are underway to provide an alternative treatment for diabetes. For example, researchers were able to differentiate embryonic stem cells into insulin producing cells to provide larger quantities of pancreatic beta cells from the embryonic stem cells. And then um, there are latest research described a translational process of generating human embryonic stem cell derived cardiac progenerator cells to be used in clinical trials of patients with severe heart failure. And the other thing is drug discovery. Beside becoming an important alternative to organ transplantation, mm, Mm, embryonic stem cells are also being used in a field of toxicology and as a cellular screens of uncover new clinical entities that can be developed as a small molecule drugs. Studies have shown that the cardiomyocytes derived from the embryonic stem cells are validate in vitro models to test the drug responses and predict toxic profiles. Um, embryonic stem cells derived from the cardiac myocytes have been shown to respond to pharmacological stimuli and and hence can be used to access cardiotoxicity and uh, the embryonic stem cell derived hepatocytes are useful models that could be used in the preclinical stages of the drug discovery. Another thing is a model of genetic disorders, and several new studies have started to address the concept of modeling genetic disorders with embryonic stem cells, either by genetically manipulated the cells or more recently by deriving the diseased cell lines identified by prenatal genetic diagnostics. And modeling genetic disorders is something that has been accomplished with the stem cells. This approach may very well prove valuable at studying disorders such as Fragile X syndrome and cystic fibrosis and other genetic maladies that have no reliable model system. And there will be a study by Yuri, a Russian American medical researcher. He developed prenatal diagnosis testing methods to determine the genetic and chromosomal disorders a month and a half early than standard and prospective parents, especially couples who have a history of genetic abnormalities or where the woman is over the age of 35 years, where the risk of genetically related disorders is high. And the other one is the repair of DNA damage and differentiated somatic cells, the embryonic stem cells, use different strategies for dealing with DNA damage. For instance, human foreskin fibroblasts, one type of somatic cell, use non-homologous end joining as the primary pathway for repairing double stand breaks during the during the cell cycle stages. Because of this error prone nature, the non-homologous end joining ends to produce mutations in cells cloning descendants. And embryonic stem cells use a different strategy to deal with all the cell types of an organism, including the cells of the germline. Mutations arising in embryonic stem cells due to faulty DNA repair are a more serious problem than a differentiated somatic cells. Thus, the mouse embryonic cells predominantly use high fidelity homologous recombinational repair. This type of repair, depending upon the interaction of two sister chromatids formed during the S phase and present together during the G2 phase of the cell cycle, that can accurately repair the homologous recombinational repair in one sister chromosome by using the intact information from the other sister chromosome. Cells in G1 phase of the cell cycle, after the metaphase, a cell division, but prior to the next round of the replication, have only one copy of the each chromosome. that is sister chromosomes aren't presented, a mouse embryonic stem cells lack G1 checkpoint and do not undergo cell cycle arrest upon acquiring DNA damage. Rather, they undergo apoptosis in response to the DNA damage. Apoptosis can be used as a fail-safe strategy to remove cells with unpaired DNA damage in order to avoid mutation and progression to the cancer. When coming to the clinical trial, phase one clinical trials for transplantation of these embryonic cells of a cell type brain and spinal cord. Injured individuals received approval from the US Food and Drug Administrative Department FDA, marking it the world's first human embryonic stem cell human procedures. And if everything went well, it was hoped that it would lead to future studies that involve people with more severe disabilities. The markers of the stem cell therapy, Geron Corporation estimated that it would take several months for stem cell lines cells to replicate. And the California Institute for the Regenerative Medicine, CIRM, is the largest founder of stem cell related research and development in the world. And other one is ASTOPC1 is a population of cells derived from the human embryonic stem cells that contain dendrocyte progenerate cells. Those are OPCs, and their mature derivatives called oligodendrocytes provide critical function support for nerve cells in the spinal cord and brain. Um, um, Asterias recently presented the results for phase 1 clinical trial testing of a low dose of AST-OPC1 in patients with neurological complete thoracic spinal cord injury. The results which showed that AST-OPC1 was successfully delivered to the injured spinal cord site. Immune monitoring of subjects through one of the post-transplantation showed to evidence of antibody-based cellular immune response to AST-OPC1 in four of the five subjects. Serial MRI scannings performed throughout the 2 to 3 years follow-up period indicate that reducing spinal cord cavitation may have occurred, and that AST-OPC1 may have had some positive effects in reducing spinal cord tissue deterioration. Concerned the controversy, adverse effects. Before going to the ethical debate and adverse effects, here I want to um, completely showing the stem cells. Just now we discussed, and we already discussed about the features of the stem cells and our classification of the stem cell, and it has the self-renewal potency. And see here how to self-renovate the stem cell differentiation into the mature cell, that is called differentiation. And again self-renewal taken place by the stem cell. What is potency here is a potency specifies the differentiation potential the potential to differentiate into the different cell types of the stem cell. Potency is classified into totipotency, pluripotency, multipotency, and unipotency. Here you can see when the sperm comes into egg called as fertilization. Here you can see the fertilized egg and totipotent stem cells. Here you can see, and the blastocytes containing the pluripotent cells means multipotent cells, um, uh, pluripotent cells, and isolated pluripotent cells from inner cell mass. Here the culturing of the cultured pluripotent cells you can see here. Uh, and what is types of potency? Totipotency, stem cells. Stem cells are found only in early embryos. Each cell can form a complete organism within 1 to 3 days. And pluripotent stem cells exist in undifferentiated inner cell mass of the blastocytes in 5 to 14 days and can form any of the above 200 differentiated cell types found in the body. Stem cells can form most or all cell types in the adult, and multipotent cells, stem cells are derived from the fetal tissue, cord blood, and adult stem cells. These cells are differentiated but can form a number of other tissues. Stem cells can form multiple types of cells and tissue types. Unipotent, able to contribute to only one mature cell type, but have the property to self-renewal, which distinguishes them from the non-stem cells. As just we discussed, embryonic stem cells are derived from the 4 or 5 day old human embryo that is in the blastocyst phase of the development. They have the ability to form virtually any type of cell found in human body. Embryonic cells are pluripotent and give rise during the development to all derivatives of the primary germ layer, ectoderm, endoderm, and mesoderm that can develop in each of the more than 200 cell types of the adult body. They do not contribute to the extra embryonic membranes or the placenta. See here, in the day 1 fertilized egg, a day 2, it is divided into two halves, two-celled embryo, multi-celled embryo in day 3 to 4. Here in the day 5 to 6, blastocyst, the divisions happen and the production of blastocyst taken place. A day 11 to 14, tissue differentiation started. And sources of embryonic type stem cells, embryos and fetuses. Embryonic stem cells are obtained by harvesting living embryos, which are generally 5 to 7 days old. The removal of embryonic stem cells invariably results in the destruction of the embryo. Fetuses, another kind of stem cell called an embryonic germ cell, can be obtained from either miscarriages or aborted fetuses. Here you can see how we can isolate the embryonic stem cell lines from the mouse, not from the human. The majority of the embryonic stem cells lines are mouse, not the humans. The basic protocol for generation of embryonic stem cells is similar for all species. These embryonic stem cells are pluripotent stem cells generated from the early stage of the embryo. Here you can see the early cleavage of stage embryo and cultured blastocyst, then isolated inner cell mass and cells are dissociated and replaced and establishment of the embryonic stem cell culture. Here you can see cells are harvested from blastocyst 4 to 5 days post fertilization. The outer cell layer of the blastocyst called the trophoblast containing a, fluid filled cavity, the blastocele, and an inner cell mass of the 10 to 20 cells. The inner cell mass, which is also called the embryoblast, is removed from the culture. The cells from the inner mass are replaced in culture, and those that are viable are expanded. What are stem cells? Adult stem cells have been found in brain, bone marrow, blood vessels, skeletal muscle, skin, teeth, heart, gut, liver, and other organs and tissues. Adult stem cells can be isolated from the body in different ways, depending upon the tissue. Blood stem cells, for example, can be taken from the donor bone marrow, from blood in the umbilical cord where a baby is born, or from a person's circulating the blood, mesenchymal stem cells, which can make bone and cartilage, fat, fibrous connective tissue. Neural stem cells, which form the brain's three major cell types, have been isolated from the brain and spinal cord. Here you can see, from the body, from the amniotic fluid, and from other adult stem cells. Here the primary mouse embryonic fluid freezed. Uh, uh, First we have taken the embryonic cells here, and expanded, and here primary embryonic mouse embryonic fluids freezed and thawed and treated with the mitomycin C or UV radiation, and mitotically inactivated mouse embryonic fluids are obtained. While culturing of the stem cells, the culture conditions and types of media used for the stem cell culture depending upon the type of stem cell. There are a wide range of protocols and the products available for both maintaining the stem cells in an undifferentiated state, and for differentiating them into different lineages and the cell types. Feeder cell layers, all the embryonic stem cells cultures are maintained on feeder cell layers. Inactivated mouse embryonic fibroblasts are used as a feeder layers that provide factors and a substrate that allows the embryonic stem cells to grow and divide. There are several problems associated with mouse embryonic fibroblasts. Um, Mouse embryonic fibroblasts, including the potential for the introduction of mouse-derived infectious agents, undesirable protein transfer. And what is the methodology for culturing the stem cells? All the embryonic cell cultures are maintained on a feeder cell layers. Inactivated mouse embryonic fibroblasts were used as feeder layers that can provide factors and a substrate that allows ESCs to grow and divide. These several problems associated with the mouse embryonic fibroblast, including the potential for the introduction of the mouse derived infectious agents, undesirable protein transfer. These mouse embryonic fibroblasts can be purchased or freshly generated in the lab. The fibroblasts from 14 to 15 years old fetal mice are cultured and expanded for 3 to 4 days to be used as a feeder layer And mouse embryonic fibroblasts must be mitotically inactivated either by irradiation with the UV light or incubation with the mitomycin. C. Stalks of mouse embryonic fibroblast cells can be frozen either before or after inactivation. How does cell therapy works? Stem cells can be used to generate healthy and functioning specialized cells which can then replace diseased or dysfunctional cells. It is similar to the process of organ transplantation, only the treatment consisting of transplanting cells instead of organs. More than 80 diseases can be cured using the stem cell therapies: Alzheimer's disease, Parkinson disease, spinal cord injury, heart disease, severe burns, and diabetes. This is about uh, the stem cells and their applications. And next, coming to our another topic, in vitro fertilization, and another topic is in vitro fertilization and embryo transfer. What is in vitro fertilization? Before going to uh, the depth into the topic, first we have to know the structure of female reproductive system. Here you can see it is looking like a T shape. Here the as everyone knows, um, here the ovaries are presented, and here the fallopian tube located, and this is uterus, and this is endometrium, and this is vagina, and this is ovary. And when coming to the female reproductive system, this is colon and bladder and vagus difference and pubic bone and seminal vesicle, a prostate, urethra, a penis and epididymis, a testicle, a scrotum, corpus gland, here you can see, and anus, that is rectum also. Uh, the internal part of the scrotum, vas difference and epididymis, testis and seminiferous tubules. In case of normal fertilization, um, here you can see a number of here you can see here the ovum presented, a number of sperms get associated, get interacted with the ovum. One sperm can attach to the um, ovum, the fertilization takes place, and here you can see how the um, sperm comes for the fertilization, and here also you can see the a small picture which depicted us about the fertilization process. In natural phenomena, fertilization taken place with ovum plus sperm. But in case of in vitro fertilization, whichever the process here we discussed normally happens, this is the gynec system of the female reproductive system, and when time comes, the egg releases in the female reproductive organelle, and when the male reproductive organelle system provides the um, sperm that get attached to the um, ovum, a successful fertilization taken place. But in case of in vitro fertilization, all the natural phenomena happens in the glass, in vitro, inside the glass. So how it is happening? The basic principle of the in vitro fertilization means the hormonal treatment of female and the harvest of the ovum taken place, the mature ovum um, um, taken into the test tube, and the other hand, the natural ejaculation of the male semen taken place. That the collected male semen with the motile sperms, those also mixed in a test tube which already contains the matured and active ova, and keep it to develop embryo and transfer to the uh, um, mother. Here, once again, a basic principle of in vitro fertilization, the hormonal treatment for the production of the ova to the artificial hormonal treatment for the female, that leads to the production of the ovum, healthy ovum that can be harvested. The harvested ovum, the matured ova, is collected in a test tube, matured ova. In the other hand, natural ejaculation of the male collected semen with the motile sperm, both of them are mixed in a test tube, keep to the development of the embryo, transfer to the mother. How we will transfer that to the mother means here, as just we discussed about the female reproductive organelle, it contains the uterine cavity and oviduct. This is more important for us now. So we once the a male spermatophore is produced, then the female ovum is injected here, injected to the oviduct through the stimulated ovaries, and egg is collected from the uterus with the help of injection, and sperm egg embryo transfer. What are the hormonal treatments we give to the female and male? First one is clomiphene citrate, clomida, serophenia, and human menopausal gonadotropin, HCG, and gonadotropin releasing hormone, GnRH, analog called leuprolide and leuprona. Most of these drugs may be used alone or in a combination with other drugs. Egg harvesting. We can see before harvesting the egg through the ultrasound guided aspiration and laparoscopy. Here you can see this is the ultrasound laparoscopy. oocytes with granulosa cells and naked oocyte, 8-celled embryo for transfer, blastocyst for transfer, and implantation takes place. And 14 days after the initial cell division, it contains connecting stalk, primitive streak, and epiblast. And here you can see the viable fetus, uh, which connects to the mother with placenta, umbilical cord, and after the birth. Uh, what are the alternatives of the in vitro fertilizations? Gametic intrafallopian transfer, that is GIFT, is similar to the in vitro fertilization technique. It is used when a woman has at least one normal fallopian tube. Eggs are placed in this tube along with men's sperm to fertilize there. And zygote intrafallopian transfer, GIFT. A GIFT is a tubal embryo transfer in which a woman eggs are taken from her ovaries, are fertilized in the laboratory, and put back into the fallopian tubes rather than the uterus. And assisted fertilization techniques, when not enough sperms are available or sperm quality is not sufficient to fertilize, including the following things like partial zonous dissection and subzonal dissection, sperm injection, and intracytoplasmic sperm injection, and embryo cryopreservation, that is a frozen fertilized egg and embryo. What are the other techniques like ICSI, which is stands for um, ICSI, which is stands for intracytoplasmic sperm injection. This process is used to inject a single sperm into each egg before the fertilized eggs are put back into the woman's body. The procedure may be used in the male, has a low sperm count. This is the cryopreservation techniques in the liquid nitrogen, cryopreservation of ova, sperm, and embryo taken place. Risk, what are the risks? Superovulation stimulates eggs development, and ovarian hyperstimulation syndrome we can see as a risks. And there may be a failure to recover an egg. Ovulation has occurred, one or more eggs cannot be recovered. Pre-existing pelvic scarring or technical difficulties prevent safe egg recovery, and follicles also sometimes damaged during the treatment cycle. The eggs that are recovered may not be normal, and there might be a insufficient semen to attempt fertilization of the recovered eggs, because the man is unable to produce a semen specimen because the specimen contains an insufficient number of sperm to attempt fertilization, because the laboratory is unable to adequately process the specimen provided, or because the option to use a donor sperm as a backup was declined. A fertilization of eggs to form embryos may fail even when the eggs and sperm are normal. The embryos may not develop normally or may not develop at all. Embryos that display any abnormal development will not be transferred. And embryo development transfer into the uterus may be difficult or impossible, or implantations may not occur after the transfer, or the embryos may not grow or develop normal after the implantation. Any step in the in vitro fertilization process may be complicated by unforeseen events such as hazardous, catastrophic weather, equipment failure, laboratory conditions, infections, and human error, and so and so. Normal results. Successful rates varied widely between the clinical and between the physicians performing the procedure. Implantation does not guarantee a pregnancy. Therefore, the procedure may have to be repeated more than once to achieve pregnancy. However, success rates have improved in recent years up to 20% in 1995, 27% in 2001. Abnormal results and ectopic or multiple pregnancy may abort spontaneously, that may require termination if the health of the mother is a risk, and the number of multiple pregnancies has decreased in recent years as technical advancements and professional guidelines have led to implanting of fewer embryos per attempt. Ethical issues: bypassing the natural method conception, the creation of life in the laboratory, a fertilization of more embryos that will be needed, a discarding of excess embryos, unnatural environment for embryos, use of untested technology, not affordable for many, a mislocation of medical resources, a creation of embryos that freezing them and keeping them in limbo, exposure of embryos to unnatural substances, destruction of embryos in research, a potential to create embryos for medical purposes, a potential to select embryos, potential to modify embryos, a facilitation of the idea that embryos are commodities, a financial rewards for in vitro fertilization. Doctors dissuade them from recommending other methods to the couple. Infertility is treated as a disease and not as a symptom of underlying medical problems. Separating the traditional mother father as a model, pregnancy past menopause, religious objections. This is about in vitro fertilization techniques, and this is about in vitro fertilization. And our next topic is embryo transfer. Here you can see this slide. See this slide. And this slideshow is a general overview of the embryo transfer in beef and dairy cattle. It includes a brief discussion of the benefits and instructional methods of embryo transfer, which is expensive and costing around $1,200 for supervaluation, ovulation, insemination, embryo collection, and embryo transfer. Here you can understand the embryo transfer is a complicated procedure
with a very high difficult levels. Embryo transfer should only be performed by trained professionals. What are the objectives? Here I can outline the benefits of embryo transfer and procedure of embryo transfer. Here, this is the embryo anatomy. We already discussed the place of ovary and body of uterus, and this is vagina and vestibule. Uh, and what is embryo? An embryo is an egg that has already been fertilized by a sperm cell. It is an organism in the early stage of the development. What is embryo transfer? Embryo transfer involves the removal of an embryo from a female of superior genetics and the placement of embryo into the reproductive tract of a female average genetics. The goal of embryo transfer is to obtain the maximum number of genetically superior embryos in a minimum amount of out time. What are the profits of embryo transfer? Traditionally, cows produce only one calf per year. Embryo transfer allows the production of many offsprings within a year from a single cow. And embryo transfer can increase the genetic and potential of the herd in a relative short period of time. Embryo transfer can increase milk production in dairy herds. Embryo transfer can increase weaning weights in beef and dairy herds. And this also allows other producers to take advantage of superior genetics because frozen embryos can be shipped almost anywhere. And embryo transfer preserves a superior genetics for further generations due to embryo freezing. And what are the necessary equipments for embryo transfer? Here you can see the plastic media bag and Foley catheter and embryo filter and microscope to observe straw and rod. And we also need some drugs like hormones, polytropin hormone, FSH, and uh, prostaglandin hormone, luteolase, and penicillin, lidocaine, and plastic sleeves, bull semen collected pipe. The process of embryo transfer begins with the selection of a donor cow. The donor cows will contribute the embryos to be transferred, and donor cows have superior characteristics. Here we have to characterize or we have to select a best donor cow um, for embryo transfer, which should have the following characteristics: high milking ability, high growth rate, outstanding reproductive capacity, and here bull selection taken place like this. Next, a bull with superior genetics should be selected, and breeding can occur naturally or by artificial insemination. Recipient cows must be selected. Recipient cows serve as surrogative mothers, foster mothers to the cows, but contribute no genetic information. For this reason, the genetic makeup of the recipient cow is not an important as the makeup of the donor cow. Or recipient cows, however, the recipient cow must be able to maintain her pregnancy to term and produce an adequate milk supply for her calf. It is very important to stress that A recipient cow must have good milking ability. Other genetic traits aren't as important, but the available milk supply to the newborn calf have a huge impact on calf's life. Synchronizing the estrus cycles. Um, Once the donor and recipient cows have been selected, they must be synchronized. So they are on the same phase of their estrus cycle. It is important to synchronize estrus cycles because the reproductive environment of the donor and recipients must be identical in order for the embryo to survive the transfer. Synchronization of the estrus cycle is a complicated process, and the donor and the recipient cows must be on the same phase of their cycles for the embryo transfer to be successful. Synchronizing the estrus cycle is controlled by the production and secretion of the hormones at the proper time during the cycle. The prostaglandin, PGF2 alpha, is the hormone that used to synchronize estrus cycle of the donor and recipient cows. Synchronizing the estrus cycle with the help of prostaglandin is produced naturally by the cow. However, a synthetic version of prostaglandin is utilized, is given in one or two injections to synchronize the estrus cycle, like 5 cc doses through intramuscular in the neck or hip is given, the lutalize is given to the cow, and preparing the donor cow to be flushed. Before the donor cow is flushed, she is super ovulated with a series of injections of follicle stimulating hormones, and ovulation is the process of releasing eggs. A super ovulation causes the ovary, the female reproductive organ, to produce many follicles. The word flush is introduced here. A flush is the process of removing the embryos from the reproductive tract of the donor cow, and flushing in address in later slides. Breeding the donor cow. When the donor shows signs of uterus, the time period during the estrus cycle when she will allow breeding, she is ready to breed. Some signs of estrus are riding other cows, clear vaginal mucus, and pacing the fence. Here you can see the breeding the donor cow. If usual artificial insemination, the donor cow should be bred at least twice to ensure that the eggs are fertilized. The flush. Once the donor cow has been bred, the embryos are allowed to grow for 6 days. During this time, the embryos are also travel down the reproductive tract from the oviduct, the site of fertilization, to the uterus, where they can be flushed out. On the seventh day, the embryos are ready to be removed. This process is called flushing. Here you can see embryo professionals use a non-surgical method to remove the embryos. The process requires experience and a patient steady hand. And an injection of lidocaine is given prior to the flush to reduce the pressure and stress on the donor cow and make the flush easier for the embryo transfer professional. This is an intramuscular injection given in the fatty part of the tail head. The effects of lidocaine are seen a few minutes after the injection. When the tail is limp, you can begin the flush. The flush, to begin the flush, a catheter is passed through the cervix into one uterine horn. Here you can see the catheter structure. At this point, uh, embryo transfer professionals insert one hand into the rectum and finds the reproductive tract. Holding the cervix in hand, she or he inserts the catheter into the vagina, passing it through the cervix and into the one uterine horn. A metal rod inside the catheter treats the catheter while it is being inserted. The flush continued. Here, here you can see the catheter contains a balloon that is inflated with a saline solution in order to seal the entrance to the uterus, so fluid and embryos are not lost. The uterine horn is filled with the flushed media and a message to allow the embryos to flow out the tract. This process is repeated several times in each uterine horn. A collecting of embryos are carried out of reproductive tract through their plastic tubes and collected in a filter with the flush media. The pores in the filter are smaller than the embryo, so excess fluid drains out of the filter without losing the embryos. Injecting the penicillin, the donor cow is exposed to infection during the flush due to all the foreign objectives that are put into her reproductive tract. Penicillin will kill all the infections the donor cow is exposed to, as well as any embryos that were not flushed out. The donor cow to carry a calf, because this defeat the purpose of embryo transfer. So after the embryos have been flushed out, uterus injected with the penicillin to kill any missed embryos or infections. Embryo statistics: an average of 7 to 10 embryos is collected from the each flush. However, the number of embryos obtained from a single flush may range anywhere from the 0 to 60. Separating the embryos. In the lab, embryos are separated from the flush media and examined under a microscope to determine their quality and the stage of development. Embryo size and the quality um, is measured under the microscope in the size about 0.2 mm. Under only undamaged embryos at appropriate or proper maturity should be transferred. Embryo quality. Here you can see the embryos on the left are damaged, should not be transferred, and the embryos on the right is proper maturity and quality and should be transferred. Transferring the embryos are transferred and put into a small plastic